In the spirit of CGSI, I will introduce myself. Hi, I'm Melissa Gymrek. I'm over at beautiful UC San Diego. Our lab is really interested in repetitive regions of the genome, and they have kind of a reputation for being, either junk DNA or just really difficult to deal with. So hopefully, by the end of this tutorial, I will convince you that repeats can actually be quite interesting to look at, and not all of them are all that scary. In spirit of tutorial, I came up with some objectives that we'll try to get to. The first is to provide some background on repetitive regions of the genome and why they're exciting, and also kind of how we think about developing methods to systematically analyze repeats. I'll also try to give some practical tips for if you wanted to run any of these analyses yourselves, and also a bit about kind of some of the challenges that you might run into. And then I will end by trying to introduce some interesting biological questions that we can go and ask once we have methods that actually allow us to do systematic analysis of repeats. And feel free to raise your hand and ask questions. I'll try to look out for questions throughout the talk. I'd much rather this be interactive than talk for the whole time. So as genomics people, one of the grand challenges that we are faced with is trying to go from genotype to phenotype. So a lot of us are in the business of looking throughout the genome for regions that, where we see some genetic variation between individuals, and then trying to correlate that genetic variation with phenotype. So here, if you had an A versus a T at this position, does that correlate with your disease risk or not? And collectively, as a field, we've gotten quite good at doing this. We can look at each position across the genome that is variable amongst humans, and we can ask whether your genotype at that position is correlated with some phenotype. That's what we do when we do GWAS. And we can identify many different regions of the genome that are associated with different traits. This happens to be a cool GWAS for eye color, but we could do this for many other traits as well. All right, so collectively, we found something like tens of thousands of different association signals between regions of the genome and some trait of interest. But even though we have all of these cool signals, our work is obviously far from complete. If we look at most GWASs done, even though we have lots of these kind of skyscrapers in our Manhattan plots, the number of these where we actually know what's going on, I can probably count on my hands or a couple of people's hands. And we're also still not that great at predicting someone's value for a certain trait or for disease risk from their genome. All right, and part of that might be because we don't have the whole story with the way that we have been analyzing the current data sets. So most of the time, when we go and do something like a GWAS or other types of genome-wide studies in a lot of people, most of the time, we tend to be focusing on these point mutations, or the single nucleotide polymorphisms, or SNPs. And these are places in the genome when you have a single base pair, and it can vary. So in this case, either you have an A or a G. And these are really convenient because, well, for one thing, they're really easy to go and genotype from either sequencing data or SNP array data. But they're also convenient because they are almost always biallelic. Usually there's only two possible nucleotides you could have had at each position. And that is really convenient because now we can go and just annotate each person as having, say, 0, 1, or 2 copies of the alternate allele, in this case the G, at this position. All right, so instead of letters, now we can just keep track of a bunch of 0s, 1s, and 2s and do our association studies with that. So generally, having 0s, 1s, and 2s makes a lot of the math for things work out really nicely. However, this is just one way that genomes can vary from each other. In fact, genetic variation can actually come in many different flavors. And as we kind of see many times in biology, things can always get more complicated than we originally thought they might be. So I'll just motivate by a couple examples of where the answer turned out to be something more complicated. So in fact, if we look at some of the strongest signals identified by GWAS to date, indeed the answer can turn out to be more complicated. So this is one of my favorite GWAS signals for schizophrenia. Turns out the top schizophrenia associated locus in the genome is in the MHC region. And if you zoom in to this locus and look at it in detail, it turns out to be not driven by a single causal SNP in the data set. In fact, if you zoom in, there is a more complicated structural variation going on, where there's not just 2 different alleles that control your risk, but there's these kind of 4 different structural versions of the C4 gene in this region. They vary by things like copy number of a sequence and also have some complicated retrotransposon element inside of them. And in fact, if you look at this more complicated variant, then you can much better explain the signal in this region. One more of my favorite GWAS hits. This is an example for height. So one of the top loci in the genome for height is in this region overlapping this ACAN or aggrecan gene. If you zoom in to that gene, there's actually this polymorphic tandem repeat. This is actually quite big in a coding region where there's a repeat unit of 57 base pairs here. And depending on your copy number of that repeat, that's really what is driving the association signal in this region. So again, it wasn't something as simple as there's a single SNP, and if you have an A, you have this height, and if you have T, you have this height. It's really something that has many, many different alleles that are possible, and based on your copy number there, that's what's driving variation in height at this locus. And in fact, some of the strongest GWAS hits to date that we have figured out what's going on tend to turn out to be something that is a little bit more complicated than we originally thought. Okay, so hopefully that's motivation why we need to think about different flavors of ways that our genomes can vary from one another. We will focus on one type of genetic variation, which is my favorite, but doesn't explain everything, of course. We will focus for the rest of the talk on a type of genetic variation called short tandem repeats, or STRs. STRs are regions in the genome where you have a repeat unit. In our case, we're defining this to be between 1 and 6 base pairs, where you have copies of that repeat in tandem. So in this example, we have a CAG repeat. Why are we so interested in these STRs? Well, one reason is that they're all over the genome. Depending on how you count these, there's something like 1.5 million places in your genome that are repetitive in this fashion. Most of these are falling in non-coding regions, although occasionally they can fall in coding regions as well. So that's one reason, is that they're all over the place. The other reason that we're really interested in these repeats is they tend to be highly polymorphic in copy number. Right, so mutation rates at these repeats is something like 10 to the minus 4 per locus per generation. It's several orders of magnitude higher than the mutation rate for point mutations. Right, so these are constantly kind of adding or deleting copies of this repeat unit. So if you were to look at a single STR across different copies of the chromosome in the population, maybe some people have 5 copies, some people have 10 copies, et cetera. So really, really polymorphic in that way. So despite being one of the largest sources of genetic variation, these can be more complicated to look at and require kind of different approaches. So are often kind of ignored in these genome-wide studies. You might have already heard about STRs. As a side note, STRs are actually widely used in other fields besides human medical and population genetics. Because they're so polymorphic, they're really useful in applications where you want to identify someone. So these, for example, are used by the FBI for forensics purposes. They have these 13 markers from this CODIS set, and they can use the copy number of the repeat someone has at each of these 13 markers to go and look them up in their criminal databases. They've also been used to solve fun cases like the Boston Strangler case. These STRs are also used for purposes like genealogy and paternity testing, again, because they're so polymorphic. In my distant past, we used this fact to try and re-identify some of the HapMap samples. So we know some of the names of the 1,000 genomes in HapMap samples. All right, but in this case, we're interested in STR associations with human traits. Right? And so STRs have long been known to be implicated in various different human diseases. In fact, some of the first regions of the genome where we're actually able to map what is the causal gene for this disease turned out to be these repeat expansions. So for example, in the case of Huntington's disease, you have a CAG repeat that is in a coding region. Most people have around 20 or so copies of the CAG repeat, but once it expands to 40 or more copies, you end up with Huntington's disease. One cool thing about Huntington's disease, but also some of these other repeat expansion disorders as well, is that this locus isn't acting just like a switch. You have the disease or not. Actually, the number of copies of the repeat you have can actually end up giving very different looking phenotypes. So in the case of Huntington's disease, the longer the CAG expansion you have, the more dramatic it is, the earlier onset the disease, but also the more severe the symptoms. So I've always thought this is really cool, again, because it's not just acting like a switch, it's more of like a single locus is acting kind of more like a dial, which you can control the severity or the type of the phenotype. Right, so there's something like 40 or so diseases that are caused by those type of repeat expansions. But actually, those type of expansions are relatively rare in humans, so they're rare, but they can have really dramatic effects. In fact, if we look at the majority of genetic variation at these STR regions across healthy individuals, it's much more of these more modest changes, these stepwise changes in copy number. So these tend to have more modest effects, but they're also much more prevalent across the genome and across individuals. So I just want to clarify that the methods that we're talking about today are mostly focusing on this more common kind of stepwise variation in copy number across healthy individuals in the population. These more modest changes in STR variation used to be kind of considered junk DNA or not very important. That's why they're used for things like forensics or genealogy or linkage. But in fact, we're learning more and more that even these more modest changes in STR copy number can play important roles. For example, these STRs can form things like transcription factor binding sites. Variation in STR length can influence things like nucleosome positioning. So we're learning more and more that it's not just these big expansions that matter. It's also these smaller changes in STR copy number can be important for influencing things like gene regulation. So really, what we're going to be thinking about is, we mentioned there's something like 1.5 million STRs across the genome that we haven't really been analyzing in these genome-wide studies. Now, can we think about designing methods that will allow us to integrate analysis of all these different STRs across the genome into things like our GWAS studies or other types of genome-wide studies of genetic variation? All right, doing this is going to require thinking about things a little bit differently than we're used to thinking about for SNP variants. So a major difference is just how we are going to have to encode genetic variation at these repeats. All right, so I mentioned earlier that SNPs are really convenient because most of the time they have only 2 alleles in the population. So at each SNP, we can actually just encode each person as having 0, 1, or 2 copies of the alternate allele. STRs will look a little bit different. Here, there can actually be many, many different alleles at a single locus. So for example, if this is an AC repeat, we might have 2, 3, 4, 5, etc. copies of the repeat. And now, a person's genotype, we can't just say 0, 1, or 2. What we actually want are the 2 different copy numbers they have of this repeat on their 2 different copies of the chromosome. All right, so there's many different possible alleles and also many different possible genotypes that each person could have. All right, so basically all the tools that we have that are really great, but that assume things are biallelic variants and can be coded as 0, 1, or 2, won't always just work out of the box for our applications with STR genotypes. Okay, before we go on, I just stop if there's any questions. Cool. Oh yeah, question. They're not always triplets, and I'll talk about this complication a little bit, but we tend to define them as things where the repeat unit is between 1 and 6 base pairs, but it's a fuzzy definition. Yeah?
You mentioned a mutation rate of 10 to the -4 for like the repeat sort of expanding or collapsing. Is there a sense of how often like a locus of these gets established? Like a new repeat unit? Do you understand?
So the mutation rate is kind of like an overgeneralization because it's very different depending on the properties of the STR. A lot of these get born by like retrotransposons that have an STR in them, like jumping to a new place and establishing one. But also a lot of things that would usually get classified as like indels end up copying sequence nearby and like forming a new STR region. I don't have a good estimate of what that mutation rate is, but this was for ones that are already kind of established. Cool. Okay. So I've tried to break this into two parts. The first is just looking at genotyping these things. If you were interested in profiling STR variation across the genome in a large cohort, how might you go about doing that? And then the second part, I'll talk about a couple of different applications that we could do once you had that set of genotypes for your sample. So basically our goal is to come up with something like this. So you had a large cohort of individuals. Maybe you're looking at UK Biobank, so you have 500,000 people. And you have a set of STRs that you're interested in, maybe 1 million different STRs across the genome. Our goal is to get this big matrix of genotypes. So at each STR in the genome, what is the diploid copy number for each of the samples in our cohort? Practically, there are two different ways that we tend to do this. The first is if you have next generation sequencing data, we can go and genotype these directly from the aligned reads. A lot of times we actually don't have sequencing data, and instead we just have SNP genotypes from like a SNP array, in which case we've had a lot of success using Beagle to impute these things into SNP data. But I will talk mostly about this first application here for if you actually had sequencing data. Okay, so intuitively, what we're going to do when we want to go and genotype these repeats is something like this. So first, we would come up with a list of where are all the STRs in the reference genome. And here is just an example. If you've seen these IGV screenshots before, the bottom here is the reference genome, and here we found that there is this ATT triplet repeat in this region of the reference. Once we knew where all the STRs are, then we can go in and look at all of the reads aligned to this location in each of our samples and ask, how many copies of the repeat does it look like this sample has? All right, looking at this one, we can see of the reads, each of these gray bars is reads, of the reads that span across this region, about half of them have a 6 base pair deletion, or -2 copies compared to the reference genome, and about half of them have this 15 base pair deletion, so they're missing 5 copies of this ATT repeat. All right, so that's intuitively what these tools are doing. This is CGSI, so we're going to more formally define this a little bit. So we could more formally define this by saying, okay, at each STR in the genome, we observe some set of reads aligned to the STR. We can go in and count the copy number that we observed at each of those reads and encode that in this vector R of the repeat copy numbers from each read. And now we would like to infer the diploid genotype, or just the number of copies of the repeat on each of the 2 copies of the chromosome. Right, we tend to do this using a likelihood approach, which have their pros and cons. In this case, we could write down a likelihood, which is just what's the probability that we observed this vector of reads, given that the underlying genotype was actually A copies and B copies of the repeat unit. Right, usually we would assume that each read in our data had a 50% chance from coming from each of the different copies, either A or B. And then we have some error model that I'm not telling you about that would allow us to compute the probability of seeing a particular read little r, given that it came from a copy that had A copies of the repeat. We could compute that likelihood for many different possible diploid genotypes, and then we can just output which values for A and B maximize this likelihood function. In the case in the previous slide, we might have inferred that that sample contained was diploid for 10 copies and 13 copies of that repeat. There are now many tools that do some flavor of this. So there are many existing tools out there now that will take in a list of repeats, and then they will try to estimate the copy number at each of the different repeats in some set that you give it. This is a biased list, and it's also an incomplete list. But one point that I want to make is that none of these tools is perfect and none of these is the best. They all have kind of different pros and cons. So for example, if you were interested in looking at kind of really big pathogenic repeat expansion in your data, then this tool, Expansion Hunter de novo, is probably the best bet. On the other hand, if you're interested in looking at this more kind of stepwise variation in STR copy number, then these other tools are a better bet. But even within those, there are kind of different pros and cons of each. For example, someone had a question about what type of repeat units do we consider to be an STR? Well, different tools have actually different definitions of what this should be. So some of these tools will consider things where the repeat unit is between 1 and 6 base pairs. Others will look at larger repeat units, but none of them kind of considers the entire range of repeat unit lengths. Some of these will be able to identify both these smaller changes and large expansions. Some of them will focus only on smaller changes, etc. So I'm not going to go through kind of each column of this table, but I do want to make the point that the best tool to use really depends on the application that you're going for. So most of these tools will require some set of STRs that you identified from the reference genome as input to the tool. And even that can be really challenging. And I'll just show some examples. So here we looked at reference STR sets that were identified using two different settings, just scanning the H238 reference genome for where are all the repeats in the genome. And I'm showing examples from these two tools, Hipster and Gangster. In some cases, they will agree. So in this case, both of them agreed that there's a homopolymer, so this poly-T repeat in this region of the genome. Here's an example where this repeat was only identified by Hipster and not by Gangster. And you'll notice that this is like a TAA repeat, but it's not always perfect. Sometimes there's 4 A's, sometimes there's 3 A's. All right, so Gangster thought this was kind of too messy to call as a repeat. Here's another one that was called only by Gangster and not by Hipster. Right? This one, Hipster just thought was too short. There was only 3 perfect copies of this tetranucleotide repeat. And still in other cases, the tools will identify kind of overlapping repeat, but the boundaries of the repeat will actually be different. Right? And it can even get kind of more complicated than this. Right? But even just deciding on which places in the genome are STRs that we should be looking at is something that it's really, really difficult to get agreement on and really kind of depends on the settings that you're using. Helia, who's a graduate student in the lab, went and downloaded kind of the reference STR sets that are used by some different published tools that are out there and tried to look at the overlap between the STRs that are called by these different tools. And based on what we saw in the previous slide, kind of not surprisingly, there's not a lot of overlap between the STRs that are called by different tools. Even when they advertise, we look at all the repeats that are between 1 and 6 base pairs long in the genome, for example. Right, so still a lot of work to be done, I think, on coming up with what exactly is an STR in the genome. I am going to go through an example of using just one of these tools. So I'll go through an example using Hipster. Hipster is kind of the tool that we often use as our default for a couple of reasons. So Hipster has kind of more bells and whistles than some of the other ones. It allows for multi-sample calling. It's pretty easy to use. It has the biggest set of STRs, and it also will infer sequence variants within the repeat in addition to just looking at the repeat length itself. Okay, if you wanted to go and run Hipster on your data, it's actually quite easy to run. So Hipster takes in, you can give it a list of aligned reads, either BAM or CRAM files for each of your samples. It needs a copy of the human reference genome. And importantly, it needs a reference set of where are all the STRs in the genome. All right, this is just a bed file of where to look for each of the STRs. Hipster has a published set of STRs that you can look at, but you can also give it a custom set if there is an STR that you're really interested in, but you didn't find it in the published reference set. All right, and then it will output a VCF file with all of the genotypes at each of the STRs in your reference set. I want to give you a sense of what the output looks like when you run one of these tools, because it can be very different than what we're used to when we look at SNP VCF files. All right, so this is, I pulled out the first couple of fields from an example VCF file that output by Hipster. You will notice a couple of things. So first we have one line per reference STR. So for each of those lines in our input file of STRs, we have a different line of output. Ref and alt, so usually for SNPs, these would just be a single base pair. So what's the base pair in the reference genome and what's the alternate allele that we saw in our samples? For STRs, these look a little bit different. So the reference allele is just the entire string of the STR region from the reference genome. And then the alt field is a comma-separated list of all the different versions of that repeat that we saw across all of our samples. So a couple of things to point out about that. One is that there's often many, many different alleles. So usually for SNPs, you only have one alternate allele. For STRs, we can have many different alternate alleles. You'll also notice that in some cases, the alternate alleles differ both by the length, so the number of copies, but we've also seen more and more that these can also vary by sequence. So in this example, this is a AAAC repeat, but there are common imperfections that we see. So for example, sometimes you have a G, like a point mutation, inside of the STR. And we have largely ignored these for most of our time looking at STRs, but we're actually seeing more and more that these sequence imperfections can be quite interesting when we go and look at kind of impact on phenotype. So we're starting to appreciate those a little bit more. In terms of the data that we get for each sample at each of these STRs, so we get your standard kind of genotype field, which is just saying which of the different alleles from that alt column the sample has. We also output these kind of convenient fields. So GB is just the difference in base pairs of the two alleles in that sample compared to the reference. In this case, our sample happened to be heterozygous for one allele that was -4 base pairs from the reference and one that matched the reference. And then finally, we output a quality score. I want to show what this looks like for a real STR in practice. So this is one of my recent favorite STRs, which is a CGG repeat that happens to be in the promoter region of this gene CBL. We have profiled this in samples from the UK Biobank. And this is an example of what the distribution of allele lengths across the population at a single STR might look like. A couple of things to point out. Again, it's highly multi-allelic. So the length of the CGG copy varies a lot from one person to the next. Another thing to point out is that this variation can often be highly population specific. So it might be hard to see, but we've coded the color is giving the ancestry of the different samples. At this particular STR, these shorter allele lengths are only seen in the Black population, whereas some of the longer alleles, for example, 16 copies, have only seen in Chinese, etc. So often these can be highly population specific. And then one more thing to point out about this particular example is that, again, sometimes the STRs can actually have common sequence imperfections in them. So in the reference genome, this is a perfect copy of CGG, but we've actually found, this is in the hatched portion of these bars, it's quite common to have one of these copies replaced by a TGG instead of a CGG. Again, we have found that those can have important implications when we look at associations with phenotypes. Okay, so we have our big VCF file of all of our STR genotypes across the genome. We're ready for analysis, right? Right, yes. Why not? Okay, we're not ready for analysis yet, because first we have to do some filtering and QC. And one kind of like golden rule that I want to get across, even if you don't care about repeats, is that generally we found that the more exciting the thing that you find in your data set, the more likely that it's actually just due to some sort of technical problem and you didn't do enough QC on your data or something went wrong. This is especially true for STRs that can be really messy. While some STRs behave nicely, others are just a disaster. So it's really important to do a lot of filtering and QC on your data set to make sure you actually believe it before you go and do any of these fun applications downstream. So I'm not going to talk in detail about like all of the different filters that we do once someone hands us a VCF file with STR genotypes in it. But I will mention that we have a tool for doing this. It's called Dumpster. It's compatible with hipster output, but also output from many of the other STR genotypers that were on that list. We can do things like filtering out calls that are really low quality based on things like their quality score or their coverage. It's also really important to just get rid of loci that don't behave nicely. So for example, one really important thing to check about the genotypes before going forward is, do they match our expectation of the genotype frequencies based on Hardy-Weinberg equilibrium? This is an important check that you do on SNP genotypes. It's also very important to do on the STR genotypes. All right, so I really want to emphasize that like filtering out the garbage is really important before you go and do kind of anything else with their data. One more thing that I'll emphasize. So this is really roughly paraphrased from one of my 3 PhD advisors that I had, who was Mark Daly. And he often said something along the lines of, you should spend 95% of your time doing analyses that you already know the answer for. This is a good way to make sure that your data set is pretty clean. And I'm not going to talk about the specifics of these for STRs either, but for example, if there are loci, like those CODIS loci from the FBI, where we know what the frequencies of the different allele lengths should be in the population, we can go see if that matches up with what we saw in our data set. We know general trends of STR variation from the literature. Like, for example, the longer the STR, the more variable it should tend to be across the population. If we were looking at mutations, we know that there should be more mutations generally from fathers than from mothers. So do we see that in the data set that we were given? All of these types of analyses, if we don't do any filtering, they don't look great. But after you filter the data and get rid of the low quality calls, then these known trends start to appear. So this is a really important part of the analysis pipeline whenever you're doing anything with big data sets. Okay, so now after we do all that, we're ready for the applications. But I'll just stop briefly for if there's any questions. Yeah.
So when you say there's imperfection in the sequences, right, when you have the STRs, can we actually consider those, like we have the CGG becomes TGG, can we analyze that C2G as a SNP?
You could, yes. And in fact, it often will get called as a SNP by like SNP genotyping tools. But sometimes it ends up being like messy because the alignments aren't great in those regions. But yes, that's how we think about it. Yeah.
So to which extent, if any, the fact that most of the sequencing is done using single reads limits the length of the repeats that can be recognized?
Yes, so that's a great question. So the tool that I talked about, Hipster, is really limited to things where the sequencing read can span across the repeat. Okay, and so depends on the length of the read, but like ballpark range, if the repeat is like 60 base pairs up, we don't do a good job. The vast majority of the repeats in the genome are actually shorter than that, and so we can get the majority of those. But really long, crazy things, we need either other algorithms, like other ones on that list, or we need longer reads. I'm not really talking about long reads, but one of the reasons that we have focused on Hipster is that it also basically works out of the box on PacBio Hi-Fi reads. And so, in the future, we hope to move, we hope more and more data sets will be generated using the long reads, and then that will solve a lot of our problems. Okay, so I will try to just briefly mention some of the cool applications that we can do once we have this big matrix of genotypes. So one of the applications, and this is the one that I'm motivated with, is looking for associations between genotype and phenotype. And we mentioned for SNPs, you can just encode each person that's having 0, 1, or 2 copies of the alternate allele, and then look for correlations between that and your phenotype of interest. Now we want to go and do something similar for STRs. There are multiple ways that we could have thought about associating STR length with different phenotypes. The way that we have focused on the most is kind of the most simplest thing that you could think of, which is for each person, they have 2 different copy numbers of the repeat. So we either just add those together or take the average. It doesn't matter which one. I will talk in a little bit, if there's time, about other ways that we could have thought about doing that association. All right, but for now, we've done this linear model where we just assume everything is kind of linear relationship between copy number and our phenotype. We've applied this to some fun data sets. So the first place that we have applied this is looking at gene expression. So if you find a repeat that is nearby a gene, can we find relationships between copy number of the repeat and expression of the nearby gene? We've found thousands of examples of this type of thing in the GTEx data set. We've also gone in and applied find mapping. I haven't talked about find mapping, but it's a really important part of a lot of our workflows to try and figure out which of these STRs we think might be causally driving any of these associations. And we find about 3,000 cases where we're reasonably convinced that the STR could be the causal driver of the association. Once we have all these associations, we can go and find interesting properties about them. So for example, in the GTEx data, we found that the majority of our highest confidence, what we call eSTRs, were these GC-rich STRs falling within the promoter regions of genes. Right? That was our kind of strongest signal from this eSTR analysis. We also found evidence that a lot of these homopolymer repeats might be acting by affecting nucleosome positioning. Right? And I'm not going into details for how we found those, but once you have these big data sets of associations, you can go in and test these different hypotheses about how they might be affecting things like gene regulation. We've more recently been applying this type of thing to look at associations for traits in UK Biobank. And we found a lot of cool examples, but I'll just show you one of my favorites. This is looking at a GWAS for a platelet trait, so this is platelet crit. And we've gone into each of these skyscrapers identified by performing, say, SNP-based GWAS, and we asked, could some of these actually be driven by an underlying repeat variant? All right, so here's, again, one of my favorite examples. There's a strong signal for platelet traits overlapping this gene CBL. And there's a CGG repeat in the promoter region of CBL. If we look at total number of copies of CGG versus platelet crit, we see this really nice relationship between the two. Turns out this exact repeat has actually been implicated in a more severe disorder. So if you get expansions in this repeat, that has been implicated in this syndrome called Jacobson syndrome that in some cases involves platelet-related phenotypes, so you get like giant platelets. Here we're finding that same locus, but much more modest variation among healthy individuals is causing kind of normal variation in platelet traits in the population. I mentioned that we have been mostly looking for linear associations between repeat copy number and different traits, but some of these associations are clearly nonlinear once we go and look at them. So even this CBL one, there's a linear range, but then it kind of plateaus on either end. This is a crazier looking one where we found this association between this tetranucleotide repeat and this gene CETP associated with HDL cholesterol. And we found this kind of like crazy looking shape when we looked at the association between total copy number and the trait. In this case, we actually had enough data. UK Biobank has like 500,000 people that we were looking at, where we can go in, instead of looking at the total repeat copy number, we could actually stratify out, so we could hold one of the alleles constant in each individual and then allow the second allele to vary. And we found that there's this cool nonlinear trend where there seems to be some kind of optimal number of copies of the repeat, and if you go away from that, then the trait goes down. And this is actually what's driving this kind of crazy S-shaped curve when we had added the two copy numbers together. Okay, I'm running kind of short on time, so I would just briefly summarize the other applications that I will mention and kind of see how they're kind of unique, fun things that you can do with STRs that you can't always do with other types of variation. So one is in terms of population genetics modeling. Right, we mentioned, again, SNPs, you often have just two different possibilities, and you can kind of summarize that with the allele frequency at that locus, but that's kind of the main piece of information that you have. At STRs, we often have many, many different alleles just at a single locus. So we can actually think about fitting population genetics models of mutation and selection at each individual locus, one at a time. So we had a really fun collaboration with Kirk Lohmuller, who's actually here at UCLA, where we tried to ask, could we identify STRs in the genome that are under selection by looking for places where the variation at the STR that we see across the population is much different than we would have expected under a neutral mutation model. And I don't have time to go through this method, but it's called SISTER. And in SISTER, we basically simulate a bunch of data under a bunch of different models of selection and ask which one of those gives us allele frequencies that look most similar to what we observed at each STR. And that allows us to quantify at each locus across the genome a selection coefficient for each individual STR. All right, and some really interesting STRs come out of that in terms of the ones that are under the strongest selection seem to be involved in developmental heart and brain-related phenotypes. Okay, one more cool application that is, again, kind of STR specific that I wanted to highlight is that most of our work so far has focused on humans, but we're now branching out, thanks to a collaboration with Abe Palmer at UCSD, to think about looking at these in other species. And one thing that we realized is that sometimes you can take the same STR, like the exact same STR in humans that was polymorphic, you can find that exact same STR in another species, like mouse or rat. So for example, in Huntington's, you look at that CAG repeat, there's also a CAG repeat at that location in other species as well. Here's just an example of one of our ESTRs that we had identified in humans. There was this polymorphic dinucleotide repeat in the promoter of this gene. Well, there's that same polymorphic dinucleotide repeat in the promoter in mouse as we found in humans, which is something kind of unique that you don't really see with point mutations. You can't usually find the exact same variant in a different species. So while we haven't done a whole lot with this yet, we're really excited about this idea of looking at the same STR that is polymorphic in many different species, which can kind of give us pretty fun opportunities to ask questions about causality and ask if it's associated with the same traits across different species. So just to conclude, so working with STRs can be hard, but we actually now have tools that can enable us to do some cool analyses of STRs in large cohorts. We've started to apply these to learn some cool new biology and some new STR phenotype links. I hope you'll be inspired to think about not just single nucleotide variants, but branch out to think about more complex variants and how they might be implicated in whatever trait you happen to be studying. STRs are just one example. I think there are probably many types of genetic variation that we are still relatively blind to and could really gain by thinking about how to incorporate these more complex variants into our workflows. So I will stop there. Thanks to everyone in my lab for helping with these things.